So I think both period 3 and 5, I think we're done through 13 and 14. If not, you can write those answers down and annotate your paper as needed. So, but let's go on to 15, which I think everybody is at. So, carbon cycle right here. We can see we got a lot of CO2 in the atmosphere. Only 0.04%, right? So not a lot, but that's what, running plants are using, photosynthesis. And we see for 3, that's what's happening. Carbon sink, photosynthesis. Number 2, we see incomplete decomposition. The dead elephant's not breaking down all the way and eventually turning into some type of fossil fuel. And then number 1, this dead elephant, anything dead is going to be decomposed, and that carbon is going to be released up to the atmosphere. So these tables can be kind of tricky. They confuse students. Just take your time. But 1, yeah, that's saprotropes. Remember, saprotropes, that's a fancy way of saying decomposer. Like bacteria. Something that absorbs it. And then incomplete decomposition, yeah, it's not being decomposed all the way, and that carbon, instead of being released back to the air, being trapped underground. And then 3, photosynthesis in producers, so carbon sink into the plant. So methanogens, they make methane gas. Big surprise, right? What is methane gas converted to in the atmosphere? So this is the reaction. It reacts with oxygen to make CO2 and water, H2O. That's something I guess you just have to know. That's kind of more the memorization part of it. For 17, population with no resistance may develop into a population with some resistance. What event could lead up to that? So what happens is bacteria do exchange. It's almost like a way for bacteria to reproduce sexually, share genes. And through these plasmids. So that's probably how it happened. Remember, it wasn't resistant, so it couldn't inherit it from before. It wasn't resistant before, so that's kind of tricky. Yeah, that looks like a good answer. And let's see. The enzymes, that's not a good answer because that's made by a gene, DNA plasmid. And antibiotics, don't cause mutations. They just kill those that are already vulnerable. So 18, I wrote a bunch of stuff down because you're going to want to, review these different types of living things. The different types of plants and animals that IB might ask you about. So here's asking what's going to have radial symmetry, one opening leading to digestive cavity, and that stuff having to come back out through the mouth. And that's a jelly or Cnidaria. Platy, all the rest of them have a mouth and an anus. Plus anus. Not too clear there, but that's anus. Mouth and anus. Platyhelminthes, the Latin for flat. And then kind of sounds like flat. Annelida, earthworms, a lot of little rings. Things like clams are mollusca. So go ahead, write down these are the basic, the 4 types of plant phyla you'll need to know. Bryophytes, Phyllonocida, Coniferophyta, Angiospermophyta, and the basic characteristics. And here are the animals. So go ahead and write this down. Make a note of it because this could they could throw this at you during the exam. Next we have this big cladogram. Remember, this all shows a relationship. The closer you are together, on a cladogram, the closer that relationship is. So there's two of them, and it doesn't. They don't annotate it here, but I want you to always annotate your diagrams. I know it helps me. This is the DNA cladogram. This is the protein cladogram. And it's asking, what's the reason why cladograms based on DNA is more reliable than a species based on protein sequence? So this would be more reliable. And you can see there's differences, like this very close association between crocodiles and birds is showing up in the DNA. And it's not showing up. Birds are more similar to mammals here with the protein, which isn't correct. And that's because of this thing called the DNA code is degenerate. That means 3 codons can code for many different amino acids. So several triplets can code for the same amino acid. And I wrote a little example of this down here. You can too, where you see this is the DNA code chart that we've used before, where you match up the first, the second, the third base. And you can see, look at these 6 codes: UAA, UUG, CUU, CUC, CUA, CUG. Those all code for one amino acid called lysine. So even though the DNA sequence is different, it may still code for the same amino acid, and that's called degenerate. And you see wobble pairing is also a nice term. You'll see a lot of times it's the third position that's different. Like here, UU, CU, CU all through it. It's only that third kind of matches up the first two. It's not so concerned about the last one. So I would definitely write this down. Now moving on to a neuron. Okay, an action potentials is showing you how sodium and potassium ions are interacting inside and outside of the membrane. So yeah, inside neuron is labeled, that's good. Here's the outside. And what you're seeing is the sodium channel being open and sodium rushing into the inside of the neuron. Remember, normally this is negative. Normally it's negative in here. And if you have a bunch of positives coming in, it's going to make it go positive. So I want you to be able to draw this graph right here showing resting potential is negative. And when you have depolarization, meaning when these sodium ions enter, it pops positive for just a brief millisecond, millisecond, up to 30. So that's the depolarization if you see the sodium rushing into the cell. You'll see this potassium channel is closed, and that's open during repolarization, which is the neuron regaining that negative characteristic. So sodium in for depolarization, sodium not doing anything, but potassium leaving during repolarization. Okay. And you should also know the sodium potassium pump resting state. This is how it's maintained negative, because 3 sodiums go out and 2 potassiums come in. They're both positive, right? But it's all about the difference in positives. So out here they're just going to have more positives than in here. And that's going to make it negative inside, because you just have way more sodiums, positives showing up outside than inside. Okay, so make sure you annotate this. There's always going to be a couple questions. Usually questions on that's some basic information you need to know about the neurons. Let's see, glucose. What do we got here? Absorbed through the protein channel, small intestine. Oh yeah, what characteristic prevents it from just diffusing right through? And we know that it's very difficult for polar and non-polar do not mix well. And when the phospholipids are non-polar, glucose is polar. So it's polar and therefore hydrophilic. Okay, so it likes water. It doesn't want to go through hydrophobic substances, so it stays on the outside. Unless the protein channel's open. You know, special channels, special gaps. Oops. This is my membrane. And this is a protein. It's my glucose. It'll go through that gap because it can't make it through the nonpolar phospholipids that is mostly surrounding it. Alright? So that's just knowing those chemical characteristics. Okay. Which blood vessel in the heart? So remember, review how blood flows through the heart. The right side of the heart, the left side of the heart. Coming into the atria, going to the ventricles, then being pumped either to the lungs or the body. Review all that stuff. So the lowest carbon dioxide, that'd be the pulmonary vein out of this list, because it's returning to the heart after traveling to the lungs. So it's not going to have very much carbon dioxide because that CO2 was released. And then all that oxygen is also absorbed. Okay, here's a little rundown of the other arteries as well, and why they're not as good of an answer as pulmonary vein. What secretes lipase, amylase, and proteases? Those are all digestive enzymes. These are the different macromolecules they function, they work to break down, and that would be your pancreas. You know, why your pancreas is such a vital organ in digestion. And here is some human reproduction. What promotes thickening of the endometrium, inhibits the hormone that promotes the development of the follicle wall. That'd be progesterone. And in general, you want to review these, the functions of the different hormones. So make sure you go ahead and do that. So make sure you write down these different, and also remember there's that chart where you have all the different levels of hormones from day 0 to 28, and then 14 when you have ovulation. You have those graphs. Okay, let's do one more page in this video. Let's see. Oh, here's a diabetic, non-diabetic after a meal. You can see this blood glucose levels. Untreated diabetic rising. Already the higher level, though, than the non-diabetic. Look at how long it takes for them to return to normal levels. Well, they don't even ever return after 5+ hours. But in non-diabetic, they go back down within 2 hours. So the best statement, what happens in an untreated diabetic patient is that, remember those beta cells, they're the ones that make the insulin. They're not making enough insulin because insulin is what lowers the blood sugar by sugar entering cells. So if the sugar enters the cells, that means it's not in the blood, right? Out of the blood into the cells. And oh yeah, beta cells, maybe better, beta, right there, that's better. Beta cells secrete that insulin, and that lowers blood sugar. And then remember, the alpha cells secrete glucagon, and that raises blood sugar. And remember, the liver is involved at that point because that sugar is stored as glycogen in the liver. You got about, if you exercise hard, you got about an hour worth, on average, for most people, of energy stored in your liver. All right, we'll break, and I'll start paper 2 next, or video 2.